Yeah, very good afternoon to everyone. We are going to start Vertical 1 session. And once again, on behalf of DDH-2020 Training Committee Chairman, Dr. G.N. Sastry, I welcome you all for today's Vertical 1 session. And today's speaker is Professor Rajesh Kumar Goel, and he is from Punjabi University. And he is mainly he completed his PhD in pharmacology, and his area of interest is neuropsychopharmacology, phytopharmacology, pharmacogenomics, as well as computer-aided drug design. And he has delivered various national and international conferences. He has published more than 80 research articles in his credits. With this small introduction, I request Dr. Goel to deliver the talk on systems biology and network pharmacology, a small approach to drug discovery. Sir, please.
Thank you, Dr. Nagamani, for the nice introduction. First of all, I would like to congratulate CSIR, particularly Dr. Shastri, for taking a lead to start the Drug Discovery Hackathon 2020 in association with MHRD Innovation Cell, CSIR, MHRD, AICT, PCI, and a number of other Schrodinger and Chemaxon partners to this. So this is really a nice program, a nice platform to share the ideas for accelerating drug discovery and training the budding scientists how they can come into this field of drug discovery. I'm Professor Goel, and I will be talking about today system biology and network pharmacology, a smart approach to drug discovery. I'm a core pharmacologist. I just learned few techniques and few, you can say, web services due to my collaboration with Dr. Sastry and my Russian collaborators, which were into this, you can say, in silico drug discovery. And we tried to make some peep into this area, and I like to share what we can do with these approaches. So in my talk, I will be covering process of drug discovery, a brief about it, and the role of classical and reverse pharmacology in drug discovery. Then we will be, you can say, debating on the reductionist and holistic approach to drug discovery, whereby we have evolved with the system biology and network pharmacology concept in modern medicine, and the need of computational tools in biology, why they are required and how they are affecting, how they are accelerating rather the drug discovery paradigm. Then I'll share a case study which we did on finding the vascular dementia new drug targets. So drug discovery. This is the process through which potential new medicines are identified. It involves a wide range of scientific disciplines, including biology, chemistry, and pharmacology. So I'm just leaving computer because it will be talking about later. Drugs are discovered historically to see either by identifying an active ingredient from traditional medicines or traditional plants, and by serendipity. Just like we know that there was a discovery of penicillin by chance. So why we need to discover new drugs? First of all, we should understand that the discovery of new drugs is one thing, that we want to fulfill the unmet need, and that unmet need is for those diseases where we do not have the molecules or the drugs, and we want the treatment to those diseases. We want new molecules to be discovered. Second one is we want to improvise the existing drugs with respect to their efficacy and safety. I mean to say, they should be more efficacious, they should be more selective as per the modern medicine concept, and they should have less side effects or toxicity. So this is where we are developing. If you see the national program for drug discovery and preclinical development of molecules, you see. So as I was telling, drug discovery and preclinical development, including 4 stages. First is target discovery. We have to find some target, and then we have to validate that this is the right target for designing or for hitting the treat the disease. Then we have to see the molecules which will be used for interacting with targets, and these molecules are refined based on whether they are having selectivity and efficacy, and they are not having more of the side effects, so that at later stage there should not be attrition of these drugs. Let's go a little bit to history. Classical pharmacology approach of drug discovery, or forward approach of drug discovery, we will be discussing. The concept of receptor was pharmacology's big idea. Paul and Rich gave this idea that there is a receptor, there is a macromolecule to which the molecules or the drugs, they interact and then they produce biological effect. So this idea of the effect of a drug on human body is mediated by specific interactions of a drug molecule with biological macromolecules, which may be proteins and nucleic acids in most of the cases. So this led to the scientists to conclude that we need only individual chemicals for the biological activity. So this made the beginning of the modern era in the pharmacology, whereby pure chemicals instead of crude extracts of medicine plants, which were earlier used to treat, were used. And organic chemistry people, they also synthesized a number of natural products which were isolated from biological sources, and they were also tested for the activity, and they were confirmed to drugs. So almost 70% of the molecules in the modern medicine, they have been from natural sources. Historically, if you see the substances where the crude extracts or purified chemicals were screened for biological activity without the knowledge of biological target. We were not knowing how they are going to act, but if they are active and we are able to isolate some active substance from those natural extracts, then the effort was made to identify what is the target for that particular active constituent. So this approach is totally called as classical pharmacology or forward pharmacology. And this is also another way to say it, phenotypic drug discovery, which is commonly used in different texts. This is the concept given by Paul and Rich, that is lock and key concept. That means a drug is going to bind to a receptor, and receptor is like a lock and drug is like a key, and they are going to fit into each other with specificity. They cannot be, we cannot say that one drug will be fitting into more than one or two receptors. That was the hypothesis stated, so it is very specific. And what are drugs basically? Drugs are nothing but they are the mimics of biological messengers. Your body is controlled by, you can say, wired communication and wireless communication through autopoids and hormones, and wired is through nerves. So this communication is making to regulate the body functions, and the regulation of body functions, as we know, is essential to have a homeostasis. So this homeostasis is maintained through this communication, and this communication is through, as I told you, some messengers, chemical messengers. So those chemical messengers are basically acting on the receptors. So the receptors are not for the drugs, they are basically for the endogenous chemical messengers. So what drugs are? Drugs are basically mimics of these natural ligands. Okay, so for example, this is the natural ligand, which we are showing as the original key here. And we are trying to make another, you can say, copy of this key, and this key is going to open the lock. And if it is opening, we are saying it is an agonist. And if we are putting something like this, the figure is showing the herb is put into the lock, that means because we are developing or we are giving some extract or some extract component. So that component is going to block that receptor, and now the original messenger is not able to fit into that lock. So this is acting as the antagonist. So this concept, this very concept of lock and key sets the ball rolling, and it is where we started the modern pharmacology, or we started the drug discovery. Now, as we are talking about keys and locks, and we should know what is the locksmith doing. And locksmith is making duplicate keys whenever we have lost the key, original key. Now, this locksmith is nothing but modern medicine chemists. They were producing multiple duplicate keys by hit and trial or by rational approach to get more keys to the existing locks. So that is how in the process of developing of new drugs, we were having number of keys or mimics of one disease, one drug, and that is able to treat the diseases. So this list of you can say keys was increasing day by day, and that is why Dr. Shastri in the first day said there was era of chemistry. The chemistry was at its peak for synthesizing novel molecules, which will be of making bigger libraries of molecules, which were able to be screened for the existing locks, and we are making more and more drugs for the existing locks. Later, the small molecules were synthesized to specifically target a known physiological and pathological pathway. Earlier to this, as I told you, a number of libraries were made by organic chemists, and those libraries were used randomly to screen for the drug targets, and we were selecting few drugs, few of those molecules which were having some affinity to those receptors, and they were having some efficacy too. But this approach required a number of chemicals to be synthesized and then to be screened, so a lot of chemical synthesis was required in this exercise. To reduce this, there was a rationale given that let's make small molecules which should be specific for a target and a non-physiological and pathological pathway. So this avoid mass screening of banks of stored compounds. So we were, you can say, selecting, filtering the compounds for a particular target. So cloning of human proteins made this possible. By virtue of that, the screening of large libraries of compounds against specific targets thought to be linked to specific diseases was possible. For example, we determined that this is an enzyme which is a target, this is a receptor which is a target. So this macromolecule, it can be blocked or can be agonized to have the specific effect in the body to treat the symptoms of a disease. So this is also called a structure-based drug discovery other way. And this approach was called reverse pharmacology and is more frequently used approach nowadays for rational drug discovery. So a number of targets were there, and these targets were common for many drugs. If you see in this figure, we are going to depict two things. One is distribution of drugs and drug targets. So these are the number of targets. This is the count of number of drugs. If you see, the more drugs, they will be acting on 1, 2, or 3 receptors at the most. And as you go down, you will find there are certain molecules which are acting as much as on 14 targets. So they are having such a pleiotropy. But if you see the number of drugs for each receptor, number of drugs for each receptor, so then you will find that this is the count. This count indicates that many drugs are concentrated into selected receptor classes. They're not widely distributed. They are having, so there is an interest of pharmaceutical company that this particular target is more, you can say, prone to have more, what we say, more appropriate to be targeted to treat those diseases which are more prevalent. So the returns in that case will be better to pharmaceutical companies. So they are acting in that. Again, distribution of drugs and drug targets, if you see, here we are giving you, these are the old proteins. Yellow ones are the old proteins and red ones are the new proteins. So new target innovation is also very less. If you see, in 1985, it was at peak and otherwise it was reduced, if you see. And similarly, when we are talking about the drugs discovered, there are two categories of drugs. One are jumping drugs, other are crawling drugs. Crawling drugs are also called as Me Too type of drugs, which are a copy of the earlier drugs, and jumping drugs are those drugs which are new, having a new mechanism, first in class drugs. So their number you see is also very less. So this is because the biology part was slow and chemistry part was very fast. So that disparity occurred there. And this is another, you can say, representation that if we talk about total human genomes, are about total number of human genes are about 21,000. You see, outer circle represents that, and these gene products are about 3,000, and in principle, 2,200 to 3,000 they are druggable. But if you see the approved drugs, they are only targeting 260 to 400 genes or gene products. So you see, what is the biological space that is not explored for, extent of biological space is not explored with the available chemical space. So this approach of drug discovery delivered a limited amount of new molecules. This is a limitation of this process. Finite synthetic molecular diversity was there. As the chemists were at the peak, they synthesized a number of compounds, but there's a limit. How much chemical diversity they can be in synthesized molecules. So this diversity was finite. So because of that, you did have new drugs, many new drugs. So majority of the new drugs previously targeted proteins. That was another issue. So that's why more of the type Me Too type of drugs. So there's slow rate of target innovation. So this was the reductionist approach which was going on, and this approach was not very successful. As you can see here, one disease, one target, and one drug is not sufficiently predicted, as evident from high clinical iteration rate. That usually end up with the financial loss to these companies. You see, this is the failed in drug trials, and this is the trial is ongoing. It's 2016 data. It's not very old data. These are the only few approved treatments for symptomatic relief of Alzheimer's disease. So you see, if this number is failing, definitely it is going to give a loss to the company. So this approach, reductionist approach, is not very good that way. Why it happens in these limitations? One disease, one target paradigm. Many single gene knockout exhibit no effect on phenotype. So we believe that this gene is responsible for a particular function, but when that gene is deleted, that function is not affected. Okay. So this is again tried that isolated deletion of two individual genes exhibit no effect on phenotype. So if we are choosing that these two genes are responsible for a function and we delete them separately in one gene in one and the other gene in the other animal, this deletion is also not going to affect the phenotype, but simultaneous deletion of two genes can be lethal or deleterious. So that means it is not sometimes that only one gene, one target on one disease, one gene paradigm is not useful. We have to think beyond that. That is why the focus is shifted to system biology. I'll talk a little bit later about system biology. So one target, one drug, one paradigm. So here is another issue whereby we thought that one drug is acting on a single target, and this paradigm was also failing. Why? Because even selectively designed drugs, which say we are very selective drugs, they are also interacting with multiple targets rather than one identified disease target within the biological system. So then we thought, no, we cannot say that one drug is acting on a single receptor or single target. It is having affinity for other receptors too. So it is not working in like a bullet. It is having what we say effect on number of other receptors. So these receptors, this polypharmacological approach of these receptor drugs led us to shifting the focus to network pharmacology. So holistic insight in drug discovery as compared to reductionist side was, let's view the disease process and drug action through the lens of system biology and network pharmacology. It provides fresh insight. Why? Because there's a general consensus of network approach is to understand disease interactome. Disease is not because of one gene. It is going to have interaction with other environmental factors, other protein products which are affected. So these interacting partners of disease target within biological system may reveal novel drug targets and may be helpful in designing effective multi-target therapies. So system biology, as of now, we are talking is it's useful for studying interacting partners of our disease protein within biological systems, this may reveal novel drug targets or novel pathways which are to be targeted for new drugs. At the same time, network pharmacology is a new use of existing drugs, and combination of therapies can be designed based on network pharmacology. We'll go one by one. So when we talk about system biology, when we say biology, so this field of biology is ultimately the source of knowledge and insight to develop new treatments. The more you understand biology, more better you are able to treat it or handle it. Significant changes to the healthcare system therefore cannot take place without changes to how biology is practiced. And we know that right from the introduction of modern science, we are doing experiments like physics, chemistry. We are also doing experiments in the biology, and because of those that to exploit that biology in the disease and health state, or we can use it for making new drugs or interventions to treat the diseases. So the reductionist thinking and central dogma theory of Watson and Crick had emphasized that genes alone are what we are made of. So this is the presumption that genes are the only, constituents which are required. If genes are good, we are good. If genes are bad, we are bad. This is a theory which was there. So with the greed of knowing what genes we have, there was a big experiment floated that was called Human Genome Project to map whole human genome. And it started in 1990, most of you may be knowing, and ended up in 2005, and we are having about a count of 20 to 25,000 genes as of now with us. And to our surprise, this, as the reductionist approach is there, they said that one gene, one function, so that means we are having a big body. We are having a big body, so we'll be having a very large number of genes, and we'll come to know that X gene is responsible for X effect, and if X effect is not going on, we'll go and correct that X gene and its product, and we'll be able to do it. But to their surprise, what happened? The number of genes which were found in human being were not considerably different than that is found in a nematode or a small worm. It was having about 18,000 genes. So this difference is not very, very effective in terms of the presumption that we can have specific gene and specific function and we can do it. So then people thought, what is the reason for the complexity? So complexity of the body is not because of the number of genes, it is because of something else. If you talk about gene is going to give you mRNA, mRNA is going to give you protein. Okay, so this is a simple process which we understand, but it is not that simple. The complexity starts when we are talking about not only the translating RNA, that is messenger RNA, but when we count the non-coding RNAs. So the non-coding RNAs, they give the complexity to eukaryotic life. You see, prokaryotics, one-celled eukaryotics, fungi, chordates, vertebrates, and then human. If you see this whole thing, we are the most complex we find. Okay, we are the most complex. But if you see at the bottom, at E. coli, if you see, we are having 84% coding RNA and 16% non-coding RNA. And in case of C. elegans, we are having 27 coding and 73 non-coding. But if you see Homo sapiens, we are having about 2% of coding RNA and 98% is non-coding RNA. So this makes a difference in the complexity. So what should we further think on it? So system biology rose in response to this reductionism, and the focus was shifted, and focus or attention was shifted not on just one part, that is genome, but to the complex interaction of genes, proteins, complex molecular pathways, which are all influenced by epigenetic layer. Affected by most endogenous and exogenous systems, like what we eat, what is environment outside, perhaps even what we think. So all these things, they are going to affect us And the system biology aims to develop integrated and holistic model of human system, whereby across multiple temporal and spatial scales, from molecules to molecular pathways, cells, tissues to organs, and to whole organism, we have to understand what is the organization, how they are interconnected. To this, I would like to add that, as I told you, 70% of the modern medicine that was derived from natural sources. The concept of system biology is discussed from last 2 decades or so in the modern medicine, but this is an age-old concept with almost 5,000 years old concept of Ayurveda and Siddha, whereby the body is taken as a whole. And we have, you can say, Tridosha, Vata, Pitta, Kapha. I'll not explain whole of it because it will be taken in some other part. So this particular system biology concept, it can be a, you can say, a Rosetta Stone to understand the modern system biology. Modern system biology can be taken, like if you see, there are some publications which talk about Ayurgenomics, where they have found that, for example, these Vata, Pitta, Kapha, these are the 3 Pradoshas, and if some people are Vata constitution, some are having Pitta constitution, some are having Kapha constitution. So based on this constitution, they find that their metabolism is different. So just to you can say reflect that this is showing the system functioning. The system functioning is based on you can say very subjective principles of Ayurveda, and this can be helpful for deriving the system pharmacology in this case. Another concept which is new and which is used from last decade or so, that is called network pharmacology. Here we can talk about that complexity of disease has led to recent interest in polypharmacology. Earlier we were not thinking like that. For example, we are taking NSAIDs, anti-inflammatory, non-steroidal anti-inflammatory. So these drugs, they were used for preventing inflammation in a number of inflammatory diseases for a long time, but they resulted in gastric erosion, gastric ulceration, gastritis. So then people thought, can we make some anti-inflammatory drugs which can have anti-inflammatory effect, but they do not have gastritis or gastric dysfunction. So this is how the network pharmacology was, you can say, initiated. So drugs with multiple targets can provide a superior therapeutic effect and decrease in side effect profile compared to ligands with a single target. We have seen that in the single target, you can have specificity, but if the disease is multigenic, in that case, you have to think of a network pharmacology approach to have a better treatment of that disease. So networks based on polypharmacology hold the promise of expanding the opportunity for novel targets and drug identification. However, it faces considerable challenges. What is the challenge? Because most of the medicine clinics, they are synthesizing now drugs or they're designing new drugs for a particular single target. Now, for designing drugs for more than one target, one molecule, single molecule for more than one target is a challenge. That challenge, we'll see how it's overcome by the medicine clinics. So now another thing we talk about is what is the role of computational tools in biology and how they're useful. Now I'm coming to this. If you see, biomedical literature is growing too fast. And if you see, this is what we saw in January, there's large number of, you can say, published data, PubMed counts, which is increasing day by day. You see, the information is too large. And if we do not have PubMed, we cannot search all articles at one time. I think everyone will agree to us. Or if we do not have these sort of interactive databases, it is very difficult to search the required data and filter data, which is required to us. We have a lot of garbage with that. So when we are listening to big data, which is approach, big data approaches are there nowadays, which are used for drug discovery. So we are having enormous amount of data. We talk about genome-wide association studies, we talk about proteomics, we talk about metabolomics. Number of, you can say, databases are available, which will be giving you a lot of data. So what you need, that is more important. And if we are not able to filter the data properly, then we will have the noises. That is called cacophony. Cacophony is definitely going to, you will not be able to listen the clear voice which you want. There will be more noise which will be disturbing you. So this is happening when we are talking about big data approach, and if the data is not appropriately filtered, so this problem will be there. So organization of biomedical data is required for that, and for this, for example, it is very difficult to interpret bundles and stacks of biomedical literature in the form of unorganized and unstructured data set. Just like big data is loud and noisy. So therefore, the organization of biomedical literature is required, and bioinformatics is playing a, you can say, pivotal role in getting this data organized. And this data can be, you can say, used, and the richness of this is available. This available data is useful to improve the drug discovery, through the lens of, as you know, bioinformatics. So here we need this is all the bioinformatics is all about. It collects the biomedical data. If I'm publishing a paper, this paper will be going to a website of the journal, and from there this database will be including that journal, that paper in that, and then it will be giving you such, asking you some key terms to search your paper, and you also search on the basis of that. So the storage is there, then organization of biomedical data they are doing. Then whenever you need, you can retrieve. And manipulation of biomedical data is also possible. Then modeling of biomedical data. This is where we say is the role of algorithms and other things for visualization, analysis, and prediction based on this data. And this is how bioinformatics is giving us new knowledge for making sense out of the biomedical literature. So up to this, we have seen that the forward pharmacology was there, then came the reverse pharmacology, but with this, all there was use of a reductionist approach. Reductionist approach is not useful. So then we moved on to you could say holistic approach. In the holistic approach, we are not talking about single target for multigenic disease. We are talking about multiple targets to be directed once at one time to have the management. So vascular dementia is one such disease on which we work, and it is not having any FDA-approved drug today for the treatment. And it is a syndrome. It is affecting learning, memory, and other, you can say, domains of cognitive domains, executive function, even complex attention. So identification of drug target is challenging owing to highly complex interplay between the cognitive and functional abilities of the brain with a diversity of vascular disease. So most of the risk factors are related to the vascular diseases. So these include hypertension, diabetes, hyperlipidemia, atherosclerosis, obesity, smoking, homocysteine, autoimmune disorders, and so on. Can we have some targets which can satisfy the development of vascular dementia? So the answer we try to find with the approach of, you can say, network pharmacology and system biology approach. So what is this basically? So first we will go step by step how we did it. It's a small, you can say, brief demonstration of how you can achieve this. Genotype-phenotype associations are first thing we have to understand. What type of genome changes are there which are reported for a particular phenotype, that is vascular dementia patient. Then what is the function of these genes and where they are involved, we have to understand that. And then what proteins are formed from these genes and how those proteins are interacting among each other. And this protein-protein interaction analysis, network analysis was factors like centrality index of that in cytoscape, where we can find which is the most suitable target. Ultimately, you have to go for validation of these targets, maybe in vitro and then in vivo, and then you have to go for the clinical trial. In this approach, we are using two things. We are making it system biology and network pharmacology approach is one, and we are adding it with the traditional knowledge from ancient systems of healthcare, which will be another form of reverse pharmacology, which will be used. So you see, the upper part is about system biology approach and network pharmacology approach, and the lower part, which is in the beige color, this is for the reverse pharmacology approach. Now, this reverse pharmacology approach is little different. How it is different? Difference is, I talked about structure-based drug discovery, the reverse pharmacology approach. But this reverse pharmacology approach, which I'm talking about, it was pioneered by Dr. Ashok Vaidya and Dr. Bhushan Butwal. They worked on the known effect of medicine plants, and they tried back to get their targets. What are the possible mechanisms of their action? So let's go for this. The first part is network pharmacology approach. First thing what we have to do is we have to extract the drug targets of those drugs which are used nowadays in one way or the other for the management of dyskinesia. So this is possible through different sources. You can go to drug bank, you can go to Camwell, or you can go to drug center. So for example, we are showing that we went to drug bank and we try to see what are the, for example, this is drug bank, and here you, if you will put the name of drug, you will find like donepezil. Donepezil is not what we say, as I told you, FDA is not approved any drug for vascular dementia, but for symptomatic control, we are using it. It's a ACH esterase inhibitor. So let's see. When we press this, we found that this is the drug, and these are the targets on which is it. So we added that target to our list. Similarly, we find for other drugs, and we made a big list, and we found that there were about 29 genes which are having very high score of interaction with these drugs. The second thing what we did is we tried to find out the genotype-phenotype association for the bad associated genes. And here there again, you will find number of sources. DisGeneT is there, proteome is there, diseases, disease connect, and then gene cards. You can use any of this, but we tried with the DisGeneT. And this DisGeneT is a web service which is giving you the genes associated with a particular drug. If you search, you put the name of disease, vascular dementia, for example, and now we are going to have the top 10 gene associations, top gene. Find what are these gene associations. So we find again a list of this. We added this list of genes again to this pool. So 29 here, 122 here. So total number of genes becomes 147. Now, once we have the genes, now we have to understand the gene ontology and gene enrichment. What we mean to say that where these genes are, where the product of these genes, proteins, they are participating, in which pathways, in which signaling that we have to understand. So for this, we have used the database for annotation, visualization, and integrated discovery. If you go, you can have like this one, David, and then what we did, we started the analysis. And for this, you need, which with the list of genes we have collected from the previous exercise that can be pasted here. And after pasting a list, you can find, like we have MWell gene ID in that. And then we submitted the gene list. After submitting, it will convert the format it requires. So it converted all the official gene symbols. So this is the ID count 147. All are successfully converted. So now it will submit, and now we have to choose which species we want. You can go for mouse, you can go for rat, and depending on your study, we were just having a virtual study where we have taken the data for human being, Homo sapiens, so that's why we selected Homo sapiens in case of to limit the annotation so that the interaction should be shown in the umbrella. Then in the background also we have Homo sapiens. So then we got the successful submission of gene list. Now this will be giving us the analysis of this data. So this is the analysis. So you see, we have selected one disease, and these are the different functional categories where we can find different things like gene ontology, what are the functions, and general annotations, mere sessions, pathways. These are involved. Tissue distribution is also possible where these are, which we are seeing in the blue genes. They are the genes which we have subjected for the analysis, and you can see this is a pathway. For example, PI3 AKT signaling pathway. Many genes are involved in this. You can find the number of genes involved in each of the pathway. So based on the cutoff you can give that what is the P score you are giving, you can find the best pathways which are useful for your target. So then next was we got 15 biological processes, okay, which were there, which are involved in the induction of pathogenesis of vascular dementia based on the data subjected. Then we got 20 KEC pathways. So you see the p-value is listed in that. You see one is we are having a big interactome. Out of that big interactome, we have to find small clusters or modules where we can target, or which are responsible for the disease. Or because of those module dysfunction, there can be disease. So this is a disease module detection algorithm, which is available online, freely available, and you can use it with some of the data from your side, and some of the data is available online, which you can download. So we downloaded its codes and its data, and then we used two files. One is the PPI text. This is the interactome, protein-protein interaction. And then we put seed genes in this, which we thought like 147 genes are there, which are really related to vascular dementia. Once we put these genes, then we had to run these two files in the Python with the code which is there. So this code gave us, you can say, an interactome of seed genes with that of the new genes. So this was containing about 722 genes, which was in the module. And out of this, 600 were new genes. Some of the genes were left. If you see, they were not a part of the module. Module means they should be integrated. So next to it, we go for reverse pharmacology approach, and we try to find the medicine plants which are reported for the diseases which are risk factor for the vascular dementia. In this case, if you see, we found for hypertension this much number of animals, for diabetes, for phosphodiesterase inhibitors, anti-anginal, anti-atherosclerotic, obesity, anti-anemic, anti-acetylcholesterol, anti-amyloid beta. So this list was long, and we had a lot of plants which were available in the huge portal. So this was about 573 additional Indian medicine plants which we were using. And then next step was which are the phytoconstituents which are reported for these plants. For those we found from Dr. Duke's phytochemical botanical basis. We put the name of the plant and we tried to see research. And then there were a number of compounds which were available, which is reported in different parts of, you can say, the plants, and we collected that data. And then after, we needed the chemical structure of these phytoconstituents, which were the number of resources we used PubChem to download, then you can have a file and you can store in that file. Nagamari, what happened?
We are able to hear.
But the screen is not available. Just a minute. Okay, we can start then. Okay. So then we downloaded 2D structure and of this compound. The next thing what we did is we had a library of 1972 phytomolecules. With these structures, we use the past prediction. This is a software which is funded by Russian academic lab of Professor Vladimir, and we are having, you can say, collaboration in this project with them. So we use this pass and we try to find out the mechanisms, which are there. So for 147 genes, we see that there were how many are there, the targets available in the pass prediction data set, and 34 MOA was predicted by the pass. And then next was of the 147 genes, out of 600 new genes, we got 15 biological processes, 20 KEC pathways, and after the 178 mechanism of action predicted by PAS for these compounds, we got 10 targets shortlisted for vascular dementia. So these are the 10 targets. If you see nitric oxide synthase, NNOS, brain inhibitor, glycogen synthase kinase 3, alpha and beta, bradykinin B2 receptor, PTP1B inhibitor, alpha 2 adrenergic receptor antagonist. So these were the targets which we thought can be useful, and these are the drugs which are available, which can be repurposed for this purpose for vascular dementia. This is the catiband, which we have used in our studies also to understand what is the effect of this. So we use Signor for verifying which target we have selected, whether it is useful or not. So for example, GSK3A, you see different downstream pathways, they show that they can be useful to use if it is stimulated thrombosis, neuroinflammation, atherosclerotic process, homocysteine level, memory learning, cell neuronal apoptosis, all these which are required to be inhibited. Same issue, whereby we are having this downstream pathways which are leading to the betterment of vascular dementia symptoms. So this work in detail, you can refer to these papers, which is available on net freely. And these are the two papers which we are showing where we have validated the predicted targets in the lab, whether they are useful for preventing vascular dementia or not. We have used two different experimental models. In one case, we have used hyperhomocystinemia to induce vascular dementia. In another case, we have used the chronic hypoperfusion model to induce dementia. Both with the different, you can say, pathologies, this was taken care by these targets. So that shows that there's a potential of these targets to be developed further for the treatment of this disease. So bioinformatics is a wonderful tool to enhance the pace of drug discovery through system pharmacology and network pharmacology both. It's a smarter way because the experimentation is reduced a lot. Otherwise, we have to give hit and trial to a number of things. So here we can talk about now. we are having studies which are undergoing where we are talking about selecting the medicinal components or medicinal plant components or medicinal plant itself for a combination of medicinal plants which can take care of more than one target which is selected out of the 10 targets so that it has a better effect. So with this, we can see that we can have identification of novel protein-protein interaction if we go by system biology approach, and we can have protein targets which are new blocks as far as our theory is concerned, and we can get the repurposing of existing drugs as new things, or we can find new drugs itself. But there are certain challenges with these approaches which we feel should be overcome. First thing is the data which is available in the network pharmacology, that omics data. There has to be improvement of the interactome data which is required. For example, we do not get the quantitative values. We don't get the right quantitative values for, you can say, the expressions in different disease states and in different populations. If we can get integration of that data, as you know that the computer is having a GIGO principle, garbage in, garbage out. So the quality of data which we are submitting to a machine, the output will be dependent on that. So based on that, more specific omics data will be having, more better will be the prediction results. So that is one thing where we have a challenge. We have to gather that data. For our population particularly, we are lacking that data. We need that data. Then need to integrate the structural information of chemicals with their pharmacological action in network-based approach. So, for example, we are not able to get the LD50, ED50 or IC50 doses of many chemicals which can be amplified that chemical networks or chemical space networks to the network pharmacology, how they can be effective. So that part is still pending. So linking biological features of chemical structural features into a network-based model should help in deciphering the components involved in pharmacological effect of a drug. So it will be the next paradigm in the drug discovery. I feel the budding scientists, they will be adding more data and they will have more projects so that we can have better quality data to do this. But last, I would like to acknowledge my collaborators which have worked with me and they taught me and rather they, you can say, inspired me to learn few things which are useful and which are really, you say, giving speedy results. So Professor Vladimir Poryakov and Professor G.N. Shastri, Professor Alexei Leknin and their team members all helped me, and we are working in a knowledge-based drug repurposing for rare and social diseases. Then my students which worked in the projects, in these projects and tried to generate the data, Dr. Dinesh Kawande, Dr. Tanveer Singh, and Mr. Sandeep Kumar. He is still working in PhD. Both of them are doing postdoctoral in the US. And definitely, I'm thankful to the funding agencies for providing me the grant to carry out this research work. That's all from my side. I would like to expect some questions which I can answer. Thank you very much.
Thank you very much, sir. It was a nice session. And due to time constraint, we would like to take only two questions. The first question is by Abhishek Jha, and his question is the difference between bioavailability and bioequivalence. What is the difference between bioavailability and bioequivalence?
Bioavailability is for a particular drug, single drug. So how, if we are taking, for example, 100 milligram dose, then how much is available out of that in the body after passing through different filters? For example, different routes of drug administration. If we give the drug IV, then we consider it 100% bioavailable. And if we are giving it by oral route, then part of it will be pre-metabolized before it reaches to systemic circulation. So the bioavailability will be reduced. So this is bioavailability. When we talk about bioequivalence, then we are comparing two formulations or two of the same drug, whether they are giving us the same bioavailability or not. If they are giving the same bioavailability, they are bioequivalent, otherwise they are not bioequivalent. Am I clear?
Yeah, yes, sir. Yes, sir. Sir, I'd like to move on to the next question, which is asked by Ginny D. And her question is, what is the role of non-coding gene in drug designing?
There's a lot which we can talk. We have to have a separate talk on that. So you see, microRNAs, that is the talk of the town nowadays when we talk about diseases and the treatment. MicroRNAs are the non-coding RNAs, but they are interfering in the translation of mRNA. Similarly, if you see, there is a short interfering RNA technique which is used to silence for gene silencing. That means gene is not expressed. miRNAs are doing the same thing. So miRNAs are not working on a single mRNA; they can work on multiple. So this is they are not having any coding value, but they are having their, you can say, regulatory value where they can silence the genes. So like there are now companies which have started designing microRNAs. So microRNAs itself, they are biomarkers as well as they are now existing as drugs.
Thank you very much, sir. And really, it was a very nice session about network pharmacology, as well as the application of various network and graph-based method in drug designing and drug discovery. Thank you very much, sir, once again from the DDH Hackathon training team. And I would like to inform the listeners that we will be starting the next session, that is Vertical 2 at 4:30 p.m. Kindly stay tuned. At 4:30 p.m. we will be starting the next session. Thank you once again, Dr. Rajesh.